Hi, this is Ricard. Thank you for watching this talk, where I'll provide you an overview of MOFA, which is a statistical framework that we developed to perform the integrative analysis of multi-omics data. So in the area of high-throughput genomics, most of the experiments that you perform can be summarized, from a computational perspective, they can be summarized into a set of matrices. So you have matrices that represent different groups of samples or different groups of cells, in the case of single cell data, which are denoted as different columns here. But then at the same time, we're also able to profile multiple groups of features or multiple modalities or multiple omics from the same groups of samples. And this is what I call views, and this is shown as different rows. It has different sets of matrices here in the rows. So, in your experimental design, you end up with some combination of these matrices. And, you can start by analyzing one of them at a time, but, ideally, you want to perform an integrative study of your data modalities. And there is different ways of performing data integration. The one I'll be mostly talking about in this talk is what I like to call vertical integration, which is essentially going from up to bottom. So that means doing multi-omics data integration when you get different omics from the same set of samples. So what are the challenges when it comes to doing multi-omics data integration? I think I can summarize the main ones I faced during my PhD into these 4 topics. So the first one is that when you collect data using different techniques, so different omics, they generally exhibit heterogeneous statistical properties. What I mean by that is that some data sets are binary, another data set is continuous, and combining different data matrices that are derived from, that have different statistical properties is quite challenging. So you cannot just concatenate everything and run, let's say, a PCA. A second challenge is that usually if you work with large studies, let's say patient cohorts, you typically have a large amount of missing values. And I don't mean just missing values at random, but systematic patterns of missing values where, for example, one patient might be missing an entire data modality. So you have different subsets of data modalities derived in different subsets of patients. And conventional statistical models are not designed to cope with missing data. And what this means is that you either have to impute entire data matrices, which is extremely challenging and dangerous, or you have to remove a lot of the data from your input metrics. So ideally, you want a statistical framework that is able to cope with missing data. Then another set of challenge is that if you work with large data sets, they typically contain a lot of sources of heterogeneity. So what I mean by that is that there is a lot of variation in this data set. And before doing differential expression analysis or any other type of downstream analysis, it's really useful to understand all these different sources of variation in your data and try to distinguish the ones which are capturing interesting biology from the ones that are capturing either technical sources of variation, such as batch effects, or an interesting biological variation. And once these sources of variation have been identified, then it becomes, the downstream analysis task becomes much easier. And then finally, just following up from the argument before, is that if you have very complex multi-omics data, typically the integration of this data, or at least getting a first glance on where is the interesting signal on this data set, it's really useful to use unsupervised and interpretable approaches that allow you, again, to grasp where I could have interesting signal that might be worth following up with more targeted or supervised approaches. So I think the most useful class of models that have been used for multi-omics data integration is what is called latent variable models. And the idea of this statistical framework is that you start from a very complex high-dimensional space, which you want to reduce the dimensionality of this data and bring it to a low-dimensional space. This low-dimensional space is defined by what we call the latent variables, and it typically brings you a compressed and denoised representation of the data, which is very convenient to visualize and perform some downstream analysis tasks. Much easier than working with a super high dimensional and complex gene expression space, for example. Now, how do you perform this dimensionality reduction depends on how you define this function f of x. And this is a whole field of statistics. So you can define f of x to be linear or nonlinear, probabilistic or non-probabilistic. There's a myriad of choices for f of x. And arguably the most interesting choice for f of x is to make it linear. Because if you make this linear, you reduce this problem into what is called the matrix factorization problem. And just looking at one data modality now, the idea is the following. So you have a matrix Y, which has dimensions, features, so let's say genes times samples, and then you're going to decompose this matrix into the product of two different matrices. So one matrix is what we call the latent matrix, which will contain these latent components or these factors that we also like to call it. And you see the dimensions are samples times factors. And then you have one matrix that connects the high dimensional space Y with the low dimensional space Z. And this is what we call the weight matrices. The dimensions of that matrix is features times factors, and this matrix is key to provide the interpretability to matrix factorization. Because for every feature and every factor, you get a score. So you can see, for a specific factor, you can see exactly which are the genes or which are the features that are driving that factor. And matrix factorization is the most useful and arguably the most used statistical model in biology, probably in the form of PCA. So PCA is just an instance of matrix factorization, but there's a lot of them, including non-negative matrix factorization, which is also very much used in practice. Factor analysis and other flavors of matrix factorization. Now, the challenge is what happens if you have multi-omics data, right? From the same set of samples, you have derived different data modalities. One option would be to just concatenate everything and run a standard matrix factorization. But that's not really a good idea. And there are different reasons why this is not a good idea. So one of them is that, as I was saying before, if you have different data modalities that have different that are of different statistical nature, meaning that one of them is binary or that they have different scales, different variances, then you might end up with principal components that are dominated by one of the data matrices. And another challenge is that it's very hard to interpret where are the principal components coming from. So let's say you have PC1 derived from this concatenated matrix. Is this derived from the gene expression data? Is this capturing covariation between different data modalities? It's not really trivial how to ask this or how to answer that in the context of PCA. And then another challenge is that PCA, or at least the conventional implementations of PCA, they do not handle missing information. So to address all these issues is why we developed this statistical framework, which is called multi-omics factor analysis. And I will try. I'm not going to go through the math of this. I'm happy to chat about that if you want. Just feel free to send me a message. But I will try to provide the intuition and the overview of how this model works. So first of all, I just want to say you can think of this as a multi-view or multi-omics extension of PCA. So it's an unsupervised method. That means that the only input you need to provide is your set of data matrices, properly normalized and so on. And this is shown here on the left. So each of these matrices is derived from your input data. But the most important assumption that MOFA makes, and it's really critical for the framework to work, is that these modalities are derived from the same set of samples or from the same set of sets. If they are derived from different sets of cells, MOFA just doesn't work. It exploits that information in order to perform like multi-omics factor analysis. All right, so then starting from this set of matrices Y, again, it's going to perform dimensionality reduction across all of the matrices at the same time. So it uses all information from all of the matrices to learn a set of latent factors, which are captured in this matrix set. That's almost analogous to NMF, to matrix factorization. Now you can see that what the model is going to infer, again, to connect the high dimensional space with a low dimensional space, is a set of matrices W, which are of dimensions features times factors. And same as in matrix factorization, this is the critical part that provides interpretability in the model. Because let's say that factor 1 was capturing, I don't know, differences between cell cycle between of your cells. Then by looking at these features here for factor 1, you can exactly pinpoint which are the genes that are responsible for that variation. To make MOFA work with multi-omics data, so to make a proper generalization of PCA, there is some mathematical attributes that need to be met. And essentially, this multi-omics structure is embedded or specified in the prior distributions of the model. So I forgot to mention, like, MOFA is a Bayesian model. So you need to specify these prior distributions, then a likelihood, and then you are going to end up inferring a posterior distribution for the weights and the factors. So we tell the model that this set of features are from one modality, and this set of features are from another modality, and this is specified in the prior distributions of the model. And then again, we make use of these prior distributions to encode what we call sparsity assumptions. So we make use of what is called sparsity priors. And these sparsity priors are extremely useful because it enables what is called automatic relevance determination of the factors. So it allows you to tell, for a specific factor, it allows you to say, this factor is connected to this specific modality, or this factor is connected to these two modalities, or this factor is not connected to any modality, which means it's a useless factor, and then it's like removed automatically from the model. How is this performed? Is again by providing a sparse solution. And I'll try to provide you the intuition here. So for example, let's look at factor 1. Again, let's assume that factor 1 could be cell cycle. Now you see here that for the first data modality, so for the first set of weights, you see there's a lot of non-zero values. So that means that this factor is active in the RNA expression data. In contrast, you can see that all the weights in the DNA methylation data are effectively 0. That means that this factor is not active on the DNA methylation data. By active, I mean that it's influencing, that is contributing to some like variability in the data. And again, you see that this factor is also active in the chromatin accessibility modality. But for example, you can see another factor, which is, for example, factor 5, which is not active in the RNA expression data. It's again not active on the DNA methylation data, but it is active on the chromatin accessibility data. So this is capturing some signal which is unique to chromatin accessibility. And this is really achieved. You see all the zeros you see here is achieved by the use of sparsity assumptions, which is not something that NMF or PCA have in their model, and it really helps when it comes to the interpretation of the model output. Okay, so just to wrap up on here, I just want to show you an example to again get the intuition of how this model works. So just consider now that we have a situation where you have a set of pluripotent cells and you are inducing a differentiation experiment, and they need to become something. Now, we could artificially now define a variable that we can call the differentiation state. And this differentiation state is essentially just capturing where in this trajectory each cell lies. So, the scale of these values is meaningless. It can go from -1 to 1 or -100 to 100. It doesn't matter. It's only captured. What only counts is, the relative positioning of each cell or its sample within that variable. And this variable is a latent or a hidden variable, right? Because you cannot just go and ask the cells, what's your differentiation state? Instead, what we can do is we can profile multi-omics from those set of samples. And this is what becomes our observations, right? So our aim is that, from this set of modalities that we measure from those cells, we want to try and recover what's the sources of variability in this data, which in that case is, uniquely the differentiation state. So we want to recover this latent variable from these observed variables. So now let's assume a very simplistic setup where the differentiation state is the only driver of variation in the data. And in that case, let's again assume that now the differentiation state is only driving variation in transcriptomics. And this is shown here by this gradient, where you see that the differentiation state is affecting all of the genes in the same way. If you now take these three data modalities and you feed them into MOFA, what MOFA is going to do is going to recover one factor, because there's just one source of variation in this data. So this is shown here on the matrix Z. And now if you look at the weights for that metric, you see that the weights for the RNA expression are going to be very high, whereas the weights for the DNA methylation or the chromatin accessibility are going to be very low, essentially zero. But now let's assume that the differentiation state is also driving variation in, let's say, genetics, so like modality 2. Now, again, if you feed MOFA, MOFA will just recover one source of variability, because there is just one in this experiment. But now when you look at the weights, you're going to see high weights on RNA and high weights on genetics. And then finally, it could be that if the differentiation state is a driver of variation across all of the omics, then you're going to recover something like that. So that's a very simplistic setup, right? Because in realistic experiments, you're going to get many sources of variability, not just one. So a more realistic solution looks like this, where you have 5 factors recovered from your data. And factor 1 in that case is capturing covariation across all data modalities. Factor 2 is capturing covariation between modality 1 and modality 2, whereas factor 5 is just capturing a variation which is specific to the third data modality. So in practice, the way we visualize that in MOFA is by this heat map. And I would argue that this is the most useful and the most important plot that you get with MOFA. So you can see that for every data modality, which you wrote here, and for every factor, you get a value of variance explained. So you see that, for example, factor 1 is explaining 25% of the variance in that modality, and also like 25% variance in that modality. Whereas factor 7 is explaining a tiny amount of variation in the second modality. So, the reason why I like this plot so much is because we're starting from a very complex, high-dimensional multi-omics data set. And what this plot is doing is reducing, is capturing the different sources of like variability in this data, represented in a very simple heat map. And, the next challenge now is to go and look for each one of those factors to try and interpret and find out the molecular etiology for them. And typically the most interesting factors are, of course, those that capture covariation across different data modalities. But now the question is, like, how do you interpret those factors? And this is where you are likely going to spend most of the time when you work with MOFA. The simplest approach is that, given that we have these feature weights, so the ones shown here, for every factor, we can visualize the weights, right, for each gene. So, for example, this is showing the weights for a factor 1, right? And each line here corresponds to a different gene. So if you have a very good understanding of your system, you can immediately spot, which are, by looking at the top genes, you might be able to spot what's the biological process underlying that factor. Sometimes it gets more complex than that. So you might want to do some gene set enrichment analysis, where you pull information across many genes using, some like database like Reactome or Gene Ontology. And this is an example of a factor that was capturing differences between cell cycle from different cells. Of course, rather than looking at the weights, so those matrices here, you can also look at the latent space. You can visualize your samples in the factor space. And this is what we typically do when we do PCA, right? So you plot PC1 against PC2, and then you color each of your samples based on some covariates, right? So now we can do the same approach with MOFA. The main difference is that you're going to be plotting MOFA factors now. And the advantage of MOFA factors is that they are derived from a multi-omics setup. So it could be that factor 1 is something mostly driven by the RNA, whereas factor 6 is something mostly driven by DNA methylation. So by combining all these factors potentially derived from different modalities, it might be quite beneficial and quite useful to distinguish subpopulations in your study. And of course, you can take this latent space and do a lot of things with it. You can do some clustering, you can do some imputation of like missing values, and you can do like random forest predictions to distinguish the clusters. And there's a lot of stuff you can do with this latent space. You can run, for example, TSNE or UMAPs. So that means running nonlinear dimensionality reduction methods on top of the linear dimensionality reduction space that MOFA provides. All right, so that's enough of the theory. So what I'll go through now is the main application that we work with MOFA when the first paper came out, and it's going to be the main one that we're going to be working on in the workshop. And this is a personalized medicine application. So it's using bulk and multi-omics in the context of a chronic lymphocytic leukemia cohort of patients. And this has been a collaboration with the Thorsten Zen's group, which they generated all this exciting study. And the data consists of 200 patients where we derived 4 different data modalities. On one side, we have somatic mutation data for a panel of pre-selected 69 mutations. And importantly, this data is binary. So it's 1 or 0. Then we have mRNA expression for 5,000 genes. So that's the most variable 5,000 genes. And interestingly, only for a subset of those patients. So for almost a third, for roughly a third of patients, we don't have RNA expression data for. And in that case, MOFA simply, it simply treats this data as missing value. So it doesn't try to impute, it doesn't remove any sample from the inference like procedure. It just simply ignores these observations if they don't exist. Then we also have DNA methylation. I think it was an Illumina CpG array for almost 4,000 CpG sites, and this is derived from almost all of the patients. And then finally, one of the most interesting data modalities is that we have drug response data for a panel of drugs. And this is actually derived ex vivo, so that means that they took blood out of those patients and they cultured their blood under different drug conditions, and they measured the viability of those cells under the drug conditions. So that's super interesting because this modality allows us to connect the molecular profiles, so that the first 3 data modalities, with something closer to the actual phenotype that this patient is having, and allows us for a personalized medicine approach in this data set. So again, let's just take these modalities. We do the standard pre-processing approaches that we can discuss later on, but you just feed this into MOFA. It's completely unsupervised. So the only thing you need to specify is the number of factors. And MOFA has an approach to sort of automatically infer an approximate amount of factors. But you can also specify that, I want 10, 15, 20, and this really depends on your application and the aim of your approach. But in that case, so we learn 10 latent factors for this data, and the factors are sorted by the variance explained across all data modalities. So by summing across all data modalities. And what we can see here is that factor 1 is extremely interesting because it's capturing covariation between all data modalities. Almost the same for factor 2, which is capturing covariation across all modalities except for DNA methylation data, where it doesn't seem to have any effect. And then the remaining factors are, again, potentially interesting because they're capturing variation in the drug response, and covariation between drug response and RNA expression. But for sake of simplicity, we'll focus here on the first two factors. So how would you go to interpret them? Here's where the biological knowledge comes into place. And the simplest way to interpret this, in my opinion, would be to look at the somatic mutation data. Because on one side, this view is binary, so it's quite easy to interpret. And we know that dogma of molecular biology, right? So we know that DNA is supposed to come first. So you have the DNA, you have the RNA, and you have the protein, and so on. So if you have some mutation upstream, it's likely going to affect all the downstream cascade, right? So I would start by looking at the weights of the somatic mutation. So let's do that. Let's actually plot the feature weights for factor 1 and factor 2. So on the x-axis, we can see the weight from 0 to 1. And here I'm showing the top genes or the top somatic mutations for factor 1 and the same for factor 2. So there are two genes or two mutations that really stand up, each one on factor 1 and the other one on factor 2. On factor 1, we have the IgHV status, so that's the immunoglobulin heavy chain variable region. And for factor 2, we have the trisomy on the 12 chromosome. And when I ran this model, when I started looking at this CLL dataset, I have absolutely no idea about CLL biology. But if you just Google these top markers, you see that they are actually the main clinical markers used in practice. So mostly IgHV, but like trisomy 12 is less understood, but it's clear that it has a phenotypic and clinical effect on the patients. So it's really cool that from a completely unsupervised approach, by inferring molecular profiles across multi-omics, we were able to pinpoint the main two clinical markers that are actually used in practice. So now let's actually do something else. Instead of looking at the weights, let's look at the samples. Let's visualize this latent space. So what this plot shows is like factor 1 and factor 2, and each dot here corresponds to a different patient. And the patients are colored by whether they have the IgHV mutation or not. So that's what the main driver of factor 1. So here you see blue, and here you have red. And then they are shaped if they have the trisomy 12 chromosome or whether they don't have the trisomy 12 chromosome. So that's the main driver of factor 2. You can see clear these are triangles and these are circles. And you can see that by looking at this scatter tool, now you can nicely divide patients into 4 different subgroups, right? So this is the subgroup that are IgHV negative and trisomy 12 positive, and so on. So this is exactly what we need when it comes to doing personalized medicine. We need to, a principled way of stratifying our patients and, classifying them into groups, and then try to link those groups with a phenotypic response, right? So we need to try and link those factors with their drug response like profiles. There is something also very interesting that I like to highlight from this plot, is that there is a lot of crosses in here, right? So these are patients for which we did not have somatic mutation data. That means that the clinician, they actually didn't know whether this patient was IgHV positive or IgHV negative. But now MOFA, by pulling information across all the different data modalities, is able to map those patients into that latent space. So for example, for this patient here, I'm not showing any statistics or probabilities, but it's quite intuitive that this patient is actually behaving like IgHV negative and trisomy 12 negative. So now we can go to the clinicians and say, it's very likely that this patient has this molecular profile, so you should be able to treat them with drug X, Y, and Z, right? So, this really shows, in my opinion, the advantage and the importance of doing multi-omics for studying, complex diseases like cancer. All right. Next thing we can do is we can take, if we have clinical covariates, which we tend to have when we are profiling patient cohorts, we can try to associate those factors with the clinical covariates. And in that case, the covariate we had, it was called time to treatment. It doesn't matter. And when we perform that correlation, you see that it strongly correlates with factor 1, as we expected. Surprisingly, it doesn't really correlate with factor 2, although this is actually a clinical marker used in practice. And the reason is that this clinical marker correlates with another clinical measurement that was not at the time of treatment. But what we can see also is that factor 7 and factor 8, which they don't explain a lot of variability in the data, but they are also correlated with clinical covariates. So it's likely that I would now go back into the model and try to characterize factor 7 and factor 8. And that's something that we're going to try to do in the workshop. Okay, so this wraps up the personalized medicine application. That's the main application that we're going to work in the workshop, but I also want to give the flexibility for you to choose whatever tutorial or topic you prefer. So the second one, which we have also, is the microbiome application. And this is a paper that was recently published in M Systems. And the idea is that we were able to profile multimodal information from the gut microbiome. So we had on one side bacteria, fungi, and viruses from a set of 59 patients that they were undergoing antibiotic treatment. And then we use MOFA to integrate all these different measurements. So each modality corresponded to a different kingdom of life, right? So we have bacteria, fungi, and viruses. And then the aim was, again, to be able to capture patterns of covariation between these different species and try to see which of those factors are actually correlated with clinical measurements. And then finally, we also have a single-cell genomics application. So that has been mostly the work of my PhD on trying to apply MOFA to single-cell genomics. The first one we have is from a data set that was generated using SCNMT-seq. So this is an experimental protocol that allows you to profile RNA expression, DNA methylation, and chromatin accessibility from the same cell. And it's an extremely exciting technology that we applied in the context of mouse gastrulation. So you have here, it's essentially an embryo that develops from a relatively homogeneous epiblast and then undergoes what is called germ layer formation. So that means that the epiblast transforms into the endoderm, the ectoderm, and the mesoderm. And we have these three modalities derived from the same cell. And MOFA, again, provides a way of integrating these different measurements and study, how much coordinated is the epigenome with a transcriptome for the different cell fate commitment events. It's a very nice data set, but I think it's one of the hardest applications because it's very noisy data. So if it's the first time you're working with MOFA, I would not really encourage you to work on this tutorial. And then again, in single-cell genomics, there is another application that I'm actually even more excited about, which is what is called the multi-omics technologies. And this was recently commercialized by Next Genomics, and what essentially allows you to do is profile RNA expression and attack chromatin accessibility from the same cell at high throughput. So in our previous technology, that's the one we kind of developed in the lab. It's quite limited in terms of high throughput, so you cannot profile thousands of cells. So profiling around 1,000 cells that we did, it was a huge effort and essentially a lot of money. Whereas this allows you to do it in a more affordable way. And this has been recently introduced, and we have a vignette that analyzes this data. So I would, if you want to work on the single-cell genomics application, I would encourage you to look in this one and not on the SCNMT-seq. But if you are a methylation fan, then of course you can try. All right, so that finishes my talk. I just want to say that everything, from tutorials to installation instructions to troubleshooting and contacts and stuff, you can find everything on the MOFA website. So the link is here on the top. And this is the set of case examples that we currently provide. All of this is in R. So MOFA is implemented in R and Python. It's a bit complex, right? Because the training procedure is done in Python because it's a lot more efficient than R. And but the downstream analysis is all done in R. So essentially all the vignettes we currently provide are in R. And these are the 6 different applications we have, and you should be able to freely choose which one you like, and then feel free to ask me questions and we can discuss about your specific application. And finally, I just want to say that if you're interested in the theory of those methods, I've recently finished my thesis on January, and I made my thesis publicly online on the GitHub link, which you should be able to see here. And I describe MOFA and factor analysis methods in general in a lot more detail than I can do in a talk. And I spend a huge amount of effort on writing this thesis. So if you want to read more about that, I would be very happy for you to read the thesis, and I can provide some feedback. And especially if you spot a mistake, just please just tell me and I'll be able to fix it. So with this, I'll finish my talk. I just want to thank you for your attention, and I'm looking forward for this workshop. Thank you.